how to change. Oh, there we go. Okay, so here's the outline. I'll start with a bit of background for how I got interested in this topic. And then I'll go through some examples of what I call constraint-based reasoning in biology. And I think having this many examples goes against any advice for how to give a talk. But I've chosen to do so in order to give you an impression of how common and how important reasoning about constraints are in many areas within biology. And this might be a bit surprising given how little focus there has been in philosophy of biology on this. And I'll use these examples to synthesize what I think are the kind of epistemic virtues of constraint-based reasoning. And just to avoid confusion, I will talk about constraint-based explanations. And this is a term I used in a paper from 2016. So this came out before Maglang's book, and he may have used this term explanation by constraint earlier, but I have to admit that I simply don't know enough about Maglang's work in order to adopt the same term. Since 2016, I stopped working on this topic, but in this paper that I'll mention later, we do cite his work, and I think there are a lot of similarities, but just to avoid confusion, I'll stick to this term. And then finally, I'll try to motivate a distinction between mechanistic explanations and what we call constraint-based explanation in order to make clearer how there are different explanatory aims in biology and elsewhere as a way to avoiding the problem of what we call explanatory imperialism. And this is a kind of, it's similar to physics imperialism, but this goes beyond this. It's simply when you use criteria for what counts as an explanation and impose that on another field that may have other aims. All right. But first, some background of how I got interested in this. So about 10 years ago, I was given a stipend for a PhD project on systems biology. And to get an idea of what is special about the reasoning styles in this field, I started out by comparing the content and illustrations of textbooks and scientific publications in different fields. So here are two examples of first-year textbooks. Now, the first one illustrates what in philosophy of biology is known as mechanistic explanations, where you have these mechanistic diagrams representing how specific macromolecules interact to bring about biological functions such as cell signaling or protein synthesis. And these are the kind of diagrams that I was very familiar with. I also have a background in biology, and I've read many textbooks full of such mechanistic diagrams. But if you then look at the similar introduction book to systems biology, it's quite striking how there is a lack of emphasis on the molecular details. You often find diagrams like these where you just have X, Y, and Z representing any transcription factor or gene. So here the focus is how these are organized, how they are wired together, whether these are inhibitory or activating interactions, and how they are kind of wired similar to how an engineer would construct a wiring diagram of an electronic circuit. So it's no coincidence that this paper uses notions such as design principles and biological circuits. This is very much inspired by an engineering mindset. And in my dissertation, I tried to clarify what this engineering mindset brings to biology. Some of it is also inspired by physics, and how this style of reasoning that I call constraint-based reasoning differs from traditional mechanistic heuristics focusing on decomposition and localization. So for people looking for these design principles, the kind of similar to what Eleanor just showed, the emphasis is on these law-based constraints. The strategy is to abstract from details in order to identify generic patterns and principles rather than describe specific systems. Before we go to the examples, I want to say a bit about how I use the notion of constraint. I see constraints as conditions that define a set of possibilities or causal operations, for instance, by limiting the degree of freedom of a dynamic system or by defining possible combinations of parameter values. They not only limit possibilities, but also enable certain patterns and functions to arise. For instance, if we have a ball or marble rolling down a ball, we could say that the structure of a ball constrains the movement, but it also enables a spiral pattern to be observed and to be described via mathematical equations. Similarly, Klifuga has an example in the book on biological complexity, where he says the rigidity of a skeleton is a constraint that limits flexibility, but it also enables upright movement on land. And to understand this kind of the scope of biological design, you often look at how different types of constraints interact and how they define a possibility space in this productive and limiting sense. So now we move to some of the examples. And we start with some reflections in theoretical biology. So we could say that the force of gravity presents a general constraint on organism design, but it's one that must always be understood in relation to organism size. Galilei famously described how the relation between bone structure and animal size is disproportionate because upscaling has to follow what later became known as the square-cube law, as shown here. So if you upscale a material, this could be a steel beam or a bone, the area scales with the square of the length increase, but the volume scales with the cube of the length increase. And because strength scales with the cross-sectional area of the material, this means that weight increases with a higher magnitude than the strength of the material. And Gould has a really nice essay called On Size and Shape, where he reflects on the constraints of organism size. And he, for instance, appeals to this cube law to explain why the body structure of dogs and elephants differ. So we can dress up a dog to make it look like an elephant, but if we really upscaled this dog to the size of an elephant, these legs would no longer work. It would need chunkier legs and a different angle of the hind legs. So it would look very different if it were as big as an elephant. And similarly, there is a limit to how big insects can get. This giant ant would immediately collapse if we upscaled it like the dog. And its open respiratory system also couldn't work because of constraints described by Fick's law of gas diffusion. Since the fusion of gas happens via passive diffusion, the surface area required for the system to work would be so big that this type of ant simply couldn't have any surface left for internal organs. Now, these reflections may appear merely as amusing intellectual exercises, but they have important implications both in engineering and biology. Many attempts to upscale the design of medieval churches have gone wrong because building constructors didn't consider the constraints of scaling relations. And one example is this Batalha monastery in Portugal, which collapsed twice during its construction in the 15th century. And similarly, theoretical biologists are often interested in exploring the constraints on the organism designs that are possible, so as to get a better sense of the possible evolutionary trajectories. In an essay from 26, Haldane reflects on how organism size relates to the influence of gravity. And he writes, and maybe you have heard this quote before, but I think this is really one of my favorite essays. So he writes, "You can drop a mouse down a 1,000-yard mine shaft, and on arriving at the bottom, it gets a slight shock and walks away, provided that the ground is fairly soft. A rat is killed, a man is broken, a horse splashes. For the resistance presented to movement by the air is proportional to the surface of the moving object. And he later writes, an insect, therefore, is not afraid of gravity. And this has inspired recent work where some crazy biologists with a drone have filmed what happens if you drop an ant from the top of a building, whether it's true that these shouldn't be afraid of gravity. And as you can see, the ant actually survives this. Moving further down scales to the world of bacteria, you can also find a lot of writings about the difficulties we have of understanding this tiny world. In his classical book, Darcy Thompson highlights that at this scale, we really have to bracket our macrophysical preconceptions and reflect upon what is possible in a world dominated not by gravity, but by Brownian motion, inertia, and the viscosity of and the electrical charge of the medium. This is also the topic of a famous paper by Purcell on life at low Reynolds number, where he demonstrates the differences of in the concept of swimming, whether you're as big as a man or a tiny bacteria. So the aim of this type of work is to understand what is biologically possible given the relation between different interacting constraints. And this is also important in contemporary biology, for instance, in comparative physiology, where textbooks often contain diagrams like this, showing how traits of organisms differ for different types of species, for instance, between a desert mammal and an aquatic mammal. And some of these are possible to describe via some law-like generalizations, for instance, allometric scaling relations. One example is the relation between mass and metabolic rate. And you should note here that the scale is logarithmic, and it shows how respiratory metabolism scales with body mass according to a quite simple power function. And you can plot a lot of quite different animals along this line. This is also important in medicine because these scaling relations have implications for how we upscale and downscale drug doses. So if we just linearly upscaled drug doses from test organisms like mice or rats to humans, we would risk severe overdiagnosis. So it's not just kind of intellectually amusing exercises, but it's actually something that informs about quite important relations. This comparative methodology aims to understand functional dependencies between an organism's different traits via a mapping of what Valtos calls constraints on being alive in his paper on design explanations, which he also describes as a non-causal type of explanation. Similarly, people working on comparative plant physiology often want to understand why certain morphological patterns are observed in quite different plant species and why other forms are not seen. And for this purpose, they rely on biophysical equations to construct so-called Morpheus spaces containing theoretically possible phenotypes. And these possibility spaces, they illustrate a trade-off in design, for instance, to account for both mechanical constraints and biological demands, for instance, interception of sunlight, water conservation, and so on. And the aim here is not to explain why specific plant species evolved, but rather to understand why some spaces are in this morphospace are quite heavily populated and why other spaces are empty. And I want to return to the example of systems biology that I mentioned in the beginning, this book by Yuri Alon on design principles. A lot of recent work has focused on identifying so-called network motifs in gene regulatory networks. So you should imagine that all these dots here are genes, and the relations between these are regulatory relations that can either activate these or inhibit these. Alon identified very frequent motifs by comparing biological networks to randomized networks. And motifs were then defined as those that were much more frequent in the biological network. And there are not that many. There were some network motifs that were heavily overrepresented. And the next step was then to investigate whether the specific wiring of these patterns had implications for specific types of functions, regardless of the causal details. So that is, regardless of which transcription factors or genes these instantiate. And he argued that these are actually generic dependence relation between structures and function. So this is, for instance, a coherent feed-forward loop that can function as a persistence detector in the input signal. So here you have two transcription factors that both have to be activated in order to activate production of an output. This could, for instance, be an enzyme needed to digest a certain nutrient in a bacteria's environment. And here it could be beneficial for the bacteria if noisy input signals doesn't result in synthesis of enzymes, as this would cost energy for the organism. Other types of network motifs have different functions, so there are also signal amplifiers or genetic switches. So the basic idea here is that the wiring, the organization of these patterns constrain the biological functions regardless of the causal specifics of these motifs. Such design principles are also used as design heuristics in synthetic biology. And I would recommend these two papers by Rami Koskinen on how synthetic biologists explore what is biologically possible. So this is also an appeal to not only see how actually explanations as explanatory, but also see exploration of biological possibilities as an epistemic aim on its own right. The final example considers so-called spandrels. So an older paper in theoretical biology from 79 commented on the spandrels of San Marco and the Panglossian paradigm. And these spandrels are famous for their decorative illustrations. But the point in this paper is that, similar to biological traits, we shouldn't just assume that things are always designed for the purpose that they are used for now. And in this case, the origin of these structures here would not be that there is more space to decorate, but simply that these are architectural constraints. These are supporting structures that are needed to design this kind of dome. Similarly, they argue that biological traits should always be considered in relation to possible evolutionary or developmental constraints. And modern research, for instance, in EvoDevo, study whether such patterns as those you can see here in the pictures arise because they are selected for specific purposes, or whether these could be byproducts of other processes. For instance, some of these could be the results of reaction diffusion processes that can be described via rather simple mathematical models. In evolutionary systems biology, people also investigate spandrels. For instance, whether the aforementioned network motifs could arise as a result not of selection of optimal functions, but as a byproduct of network evolution. So this is a paper where they conduct a network simulation where they have certain simple rules for copy and reconnection of nodes in a network. And after 2000 generations, this feed-forward loop motif actually appears quite frequent. These are the white dots here. Now, I don't have time to go through these examples in detail, but I can point to two papers where I comment more on these. But most important here is that there is this question of whether we should look at the level of specific circuit here, or we should sometimes zoom out and ask what are the possibilities and what constrains these development over time. In this paper here, we comment on explanatory integration challenges in evolutionary systems biology. And these are disagreements about the extent to which natural selection can explain the emergence of structures like these. And we argue that some of these explanations are actually compatible because they operate on different level of abstraction. And this is a more general point that I want to make for constraint-based explanations. There is a quite important difference between being interested in the causal operations or the evolution of a specific trait. For instance, how does this trait function? Why did it evolve? How does it maximize fitness for a specific organism? And then to ask why characteristic patterns arise in evolution in this greater space of possibilities, and to investigate what constrains this landscape in the first place. So I'll now try to synthesize some of these aspects and say a bit about what characterizes constraint-based reasoning as I see it. But before moving to constraint-based explanation, I want to flag that constraint-based reasoning is also recognized as an important aspect of mechanistic research. So it can be an important part of developing more detailed explanations. Some call this function one of providing how possible explanations or sketches for mechanistic explanations. I would caution a bit about this use of how possible explanations because, or at least equating these with sketches, because one of the points is that sometimes the possibility space is actually explanatory in its own right. But the basic idea here of what is often also used by systems biologists as constraint-based reasoning is that you want to understand a specific biological function, and you can then apply engineering methods to identify possible or even optimal structures that would realize biological functions. So you could, for instance, define an allowable solution space or possibility space by looking at an organism's, the constraints an organism has to deal with and physiological demands. And then you examine the specific traits and you develop models that can be used to develop hypotheses for how a specific function could be realized. And we've provided some examples of this in a paper in EJPS, where systems biologists use optimality modeling to develop hypotheses for experimental investigation. But the main point here of the talk here is that constraint-based reasoning go beyond this heuristic role. So in paper, in Dialectica, Nick Jones and I talked about constraint-based reasoning for both search or heuristics and explanation. So we argue that the role of what we call formal constraints, so this is mathematical representation of physical constraints, cannot be reduced to this heuristic role. They can play an explanatory role in different types of explanation, and one example would be what Bill Bechtel has called dynamic mechanistic explanations. And these are explanations that are mechanistic, they emphasize causal details, but they also emphasize the importance of quantitative descriptions, for instance, of dynamic processes. And I can point you to some examples of Bill's work where he gives constraints a quite prominent explanatory role. But still, we distinguish this from what we call constraint-based explanation, concerning both the extent to which formal constraints are prioritized or central in the explanation, but also with respect to what type of explanatory question is answered here. And this is what I will focus on. I should perhaps also mention here that what we called constraint-based explanation is kind of, it's intended as an umbrella term for different types of non-causal explanations, including this, what Lautus called design explanations, or what Philip Huneman called topological explanations. And I think we also cite a paper from by McLang from 15 or so, where he talks about some of these examples where you have mathematical constraints defining the scope of possibilities. So this is roughly what we mean by constraint-based explanation. But I'll try to illustrate this difference between mechanistic explanations and constraint-based explanations via an example. And this is an example where you have one system, but you can ask quite different questions. This is an example where you want to explain the capacity called robust perfect adaptation, which is the capacity of a system to maintain responsiveness of sensors by returning to the exact pre-stimulus value, even if activating signals are still present. And this is an important goal in engineering because it's difficult to design sensor systems that keep the same sensitivity even after they have been activated. But bacteria are actually able to to perform this function. An example is E. coli or chemotaxis in E. coli. Chemotaxis is a process where bacteria can swim towards nutrients or away from repellents or poisons via a flagellar motor. And there's been a lot of work into how they actually do this. And one question you can ask is, how actually is this capacity realized? And one answer to this, which is quite famous, is Barkay and Liebler's ODE model. So this is a model containing 14 ordinary differential equations describing the kinetics of ligand binding to some receptors here, methylation of various receptors and macromolecular complexes, and deactivation via demethylation. And this provides what Bill Bechtel terms dynamic mechanistic explanations. It provides causal details, it specifies the components, but it also provides an account of the dynamics of how this is carried out. But a different explanatory question is whether this capacity is an instantiation of a general design principle. And this is the question that was answered by John Doyle's systems biology group. And they argued that robust perfect adaptation is achieved through a principle that is known in engineering as integral feedback control. And this is what an example of what we call constraint-based explanation. And you can also see here the differences in how you would kind of represent these explanations. So the integral feedback control principle cites that when an environmental input changes, the difference between the actual input, which is U1 here, and the desired input is fed back to the system as the integral of the system error. And this becomes then an action signal for renormalization of the receptor. And this causes the system to asymptomatically track the steady state receptor activity. And I can't really go into details here, but it's interesting that Doyle's group derived this equation from the Barkay-Liebler model, and they show that this steady state expression depends only on the kinetic rate constants and concentrations of two of the involved molecules, which is giving up a lot of the detail that was in the original model by Barkay and Liebler. So I think it's interesting to note that the move here is not from an abstract how possibly model or a mechanism sketch to a more detailed how actually model. Actually, the inference goes in the opposite direction from a detailed model towards this explanation of whether this is a generic principle. What is also striking is that it's the same principle that explains adaptation in as different systems as bacteria and thermostats. Dial's group further proposes that this principle is necessary whenever you have robust asymptotic tracking, and now I quote from the quote here, it must have integral feedback as a structural property of the system. When combined with biological realizability, this may greatly constrain, on the basis of external behavior, the possible internal mechanisms that can be used to achieve the observed behavior. So they are suggesting that this is a universal principle that is multiple realized whenever we have this specific biological function, then we have an instance of integral feedback control. Now, it has been debated whether this is the only possibility, and it has also been contested. But it's quite interesting that it seems that there are very few ways in which biological systems can deal with this. So this is a paper where some other biologists try to use computer simulations to systematically search for possible network topologies that can achieve biochemical adaptation. And it turns out that in less than 400 out of more than 16,000 possible 3-node network topologies are capable of performing this. They've also done similar studies with more complex networks, and it's quite interesting that even a great sea of possibilities often converge to specific solutions that seem to work better. And these are often the ones that you find in nature. And out of these, specifically, I think it was 395, it turns out that these are actually examples of 2 generic types that are shown here. So one is a negative feedback loop with a buffering node, and an incoherent feed-forward loop with what they call a proportional node. The details here don't matter, but I have included this to kind of highlight how this, how the explanatory aim here differs from mechanistic research. And they say this also quite explicitly in their paper. They say in here, instead of focusing on one specific signaling system that shows adaptation, we ask a more general question. What are all network topologies that are capable of robust adaptation? And then they conclude, despite the diversity of possible biochemical networks, it may be common to find that only a finite set of core topologies can execute a particular function. These design rules provide a framework for functionally classifying complex natural networks and a manual for engineering networks. So here you also see the connection to synthetic biology. Okay, so the final part here is a clarification of why I think it's important to distinguish between these types of explanation. And as I mentioned, one motivation can be to avoid this problem of explanatory imperialism, where some scientists or some philosophers claim that certain models are not explanatory because they don't live up to the ideals that they have for what makes something an explanation. And I provide 2 examples here, one from each side. Melinda Fagan has a really nice paper where she describes clashing views of explanations in stem cell research, where some systems biologists inspired by physics approaches, and that I would see as examples of constraint-based approaches, kind of dismiss the mechanistic model of experimental biologists as merely providing ad hoc metaphors that don't really explain anything. There's a quote here that she also includes where they say, complicated mechanisms can neither identify the essential features of stem cells nor extract universal features that are present in all stem cell systems. By contrast, we start with a simple model containing only the essential features of biological system. And with this, we attempt to capture the universal behavior exhibited by all cell societies. As Fagan also points out, this may be a way of taking this quest for generality a bit too far, because it dismisses what is valuable in the mechanistic diagrams used by the experimentalists. We can also have imperialism from the other side. The other side could argue that only detailed models are explanatory, that the models developed by guys like these are not really explaining anything in biology. And I've taken here an example from a philosopher that I think fits with the views of many molecular biologists. And he's actually commenting on the example I just mentioned on this case of robust perfect adaptation. And he's saying, reasoning using abstract structural analogies is rightly identified as useful and sometimes powerful heuristics for schema construction. But explanation cannot stop here. Fascination with the apparent autonomy of design or design principles should be tempered by a realization that abstract principles bar us from explaining the actual workings of any given system of sufficient complexity. So for Mathiasen, something doesn't become an explanatory model until it contains the kind of details that, for instance, would allow us to distinguish between different types of bacteria or thermostats. But I also feel that downgrading the kind of work done, for instance, by John Doyle's group as simply a heuristic for mechanistic schema construction misses out on the explanatory ideal. So both of these, I think, take it a bit too far. I think it's important to recognize that what people investigating design principles or general evolutionary principles, what they're aiming for is something different. For them, the scope of biological possibility is an important explanatory target. They are interested in questions like these. Why are certain patterns, either dynamic behavior or morphology, common despite what may seem like endless possibilities of biological variation? And I think this type of question is similar to Bob Batterman's type 2 question, where he is interested in explaining why the same model can be applied to explain causally different systems, or why do causally different systems exhibit similar organizational features or behaviors? Why do certain systems converge to the same generic types of design? So I think it's important to mind the different explanatory aims. I think this is just a summary slide, but the basic take-home message here is that mechanistic explanation often focus on specific concrete biological systems and how they function. And these are very good for explaining how mechanisms differ among different species, whereas constraint-based explanations are aiming at addressing the question of why we see certain patterns across these systems. The aim is here to see what is the scope of biological design and what are the unifying features of what may appear as quite different systems. Why is it that we see certain patterns even in biology? So I think this should also make us revisit whether there is scope for generality in biology. Quite often it is said that biology is not a general science, it can only be a science of the particular, but I think this really depends on what we mean by generality. And in a previous paper, I used the notion of constraint-based generality to point to the kind of generality we can arrive at when we look for these abstract principles. It is inspired by what Nancy Nassessian termed generic abstraction, where she says it involves selectively suppressing information instantiated in a representation so as to make inferences that pertain only to the generic case. So we're asking what all triangles, harmonic oscillators, or persistence detectors have in common, rather than focusing on what is specific for a specific system. Similarly, constraint-based reasoning enables an understanding of the structural features that enable causally distinct systems, for instance, from bacteria to airplanes and transistors, to produce similar dynamic operations under a range of implementations. And it's quite important here to notice that the explanatory focus shifts from uncovering specific causal mechanisms to investigating the types of system organization that in general can realize a particular function. And I think this is where engineering and physics has a lot to offer biology. And I don't see these explanations as being in competition or as being in tension, because they operate on quite different levels of abstraction. So that is what I had prepared. Now I'll stop sharing.